Hi everyone, Holly Hood here again. We hope you're enjoying our first full day of our Closer Than Ever virtual conference. Just wanted to let you know this session is also pre-recorded. It is from a clinical research center tour we did back in May of this year. So we hope you get a chance to take a look and see all of the amazing things that they are doing at the Clinical Research Center. So without further ado, I will go ahead and pass it over to Neil.
Our new manager of operations. So will be a new face that you'll see a lot more regularly. And the other thing I just wanted to thank actually is Donna for joining us today as well. And hopefully, Donna, you get to learn a little bit about what I do and what we do here at the Chromosome 18 Registry and Research Society. So I'm going to introduce quickly Dr. Cody to you. Dr. Janine Cody started the organization 31 years ago and has really been leading the charge for the Clinical Research Center that we're celebrating 30 years this year in an event that we're going to be holding in September. So Janine kind of got involved with Chromosome 18 because of her daughter Elizabeth when she was born. And really, when the organization first started, there was very little known about kind of chromosome 18 conditions. And kind of originally, there was just a few people that were known in the world. But just this year, we've actually had almost 100 new members join the registry, which is amazing. Which actually might lead us to have one of the highest number of new members joining the organization actually since we started 31 years ago in 1 year. So, so welcome. And I'm going to hand it over to Dr. Cody and she is actually based at the Chromosome 18 Clinical Research Center, which is at UT Health San Antonio. So Janine, over to you. You guys are on mute, unfortunately, at the moment.
Oh, can you hear me now?
There we go. We can hear you now.
And with the mask, if I need to speak up, let me know, or repeat something, let me know. Welcome.
Need to move just a little bit closer to there we go. Perfect.
We're all vaccinated here. But
yeah, and there's nobody else around. This is virtual.
This is, yeah, it's virtual.
Yeah. Look out for virtual bugs.
If somebody, that's David, our cameraman, camera person, sorry. Yeah, if somebody comes, I'll pull up my mask. How about that? We are, thank you for being here. We are in the medical school at UT Health San Antonio. So if you were here in a real life, you would come in the front door and up the elevator. It's way at the end of this hall, up here to the 5th floor. All of this is labs and offices for the Department of Pediatrics. And so that's where we live. And the first thing you'd see when you come down this long hall is you would see this bulletin board that Anna's created. Families have sent in pictures to be included on our bulletin boards. I think we have 5 bulletin boards now. Here's my daughter Liz. She put hers right in the middle. So as we go through the tour, you'll see the bulletin boards and you can enjoy some of the pictures of the kids. So let's move down here and let me explain a little bit of background for those who don't know. So first of all, chromosome 18 abnormalities. I'm sure everyone's heard of Down syndrome. It's caused by 3 copies of chromosome 21, but you can have extra copies or extra pieces or missing pieces of any of the other chromosomes. Many aren't compatible with life, but those involving chromosome 18 are. And the most common of the chromosome 18 abnormalities is trisomy 18, in which people have 3 copies of chromosome 18. We have not really focused on trisomy 18 for a couple of reasons. First of all, other groups were already interested in trisomy 18. So we were more focused on the conditions not already being studied. Is by far the most common, which is why there were other groups already interested. But babies born with trisomy 18 are medically fragile. So their needs are very, very different from the rest of the group. And only 10% live past their first birthday. So that makes them very different from the other chromosome 18 conditions. However, we have really started to focus on the long-term survivors with trisomy 18. And we just did a survey for long-term survivors. And now I've forgotten the number, but it was like 250 or 240 or something responses of long-term survivors. So they're out there, even though the world thinks they don't exist. They do. And so we're starting to learn what we can about them. But the other conditions that we are interested in are, as I said, you can have all sorts of deletions or duplications of the chromosome. So you can have a deletion of the long arm. So you'll see each chromosome has a constriction, a waistband kind of somewhere on its length, and it's never exactly in the middle. So you always have a shorter arm and a longer arm. And the convention is that the short arm is positioned at the top, and it's called the P arm, and the long arm called the Q arm is positioned below. So any deletion of any size from any position on the long arm of 18q, on the long arm of chromosome 18 is considered to be 18q minus. So you could well imagine someone could have a little bitty deletion or a big one that's shown here, or a deletion up here and out of the center or up here or down here, and they wouldn't even be the same. So it's really a very heterogeneous group. By the same token, you can have a deletion of any size from any position on the P arm, the short arm, and it's 18p minus. Or individuals can have a normal chromosome 18 and the other chromosome has formed a ring because the caps called telomeres on both ends of the chromosome are missing, and those ends become sticky and they join together. And by definition, genetic material is missing from each end, but the amount of material missing from each end varies. So no two people in that group are the same at all. 18q minus and 18p minus occur in about 1 in 55,000 births, so they're ultra rare. The most rare is tetrasomy 18p, which is 1 in about 625,000 births. These individuals have 2 copies of chromosome 18 that are perfectly normal, but they have an extra chromosome, but that chromosome is made up of 2 p arms. So there are 1, 2, 3, 4 p arms, so it's tetrasomy 18p. So as you can see, this is a collection of people that can be very different. There's 263 genes on chromosome 18. We really only think about 10% of them have an impact when you have a missing or duplicated piece. And that's been a major focus of our research, is to figure out which are those key genes, because those are the only ones that we need to be able to manipulate in order to develop treatments. So that's been a major focus of what we do. We have enrolled about 700 families from all over the world. Annis made a map a year or so ago, which it hasn't changed substantially. And you can see where all the participants come from. We're very limited because the ability to speak English and understand English is key. Our consent forms are in English. Everything you do here is approved by Human Subjects Review Board. And so you get a consent form before you participate in anything, even a questionnaire. And many of and this is a lifetime longitudinal study, so we're following families or affected individuals for their whole life, because nobody knows what adult onset conditions they might have that could be preventable, or if caught early could be helped. So, and none of that's known. So that's what one of the things we're trying to find out. And many of the instruments we use, we try to, of course, because people are all over the world, focus on virtual evaluations as much as possible. And many of those instruments are only validated in English. And so you have to be able to understand English in order to do them. So while it would be easy to have consent forms translated, we do lots of interaction with families. We use, as I said, validated surveys in English, so we're really limited to English speakers. And we're also limited by the ability to get blood to us, which is a key part of enrollment, within 3 days. And I'll show you that in the lab and talk a little bit more about that when we get to the lab. So here are the people involved in this. It's myself, Dr. Hales, the medical director. He's now at Hershey, but still involved and still the physician who knows most about these conditions. The research staff include Annis Hill, who is out today. She's the main contact for anybody you need to contact at the research center. You always go through Annis. She makes sure that any of us remember to answer the email or answer the phone call, and many of the questions she can deal with. So she's the main point of contact for the program. Patty Hurd runs the lab, and I think she's not in there at the moment. She doesn't like to do, she likes to do in-person tours, but she doesn't like to do the video tours. So that's no problem. David Rupert, who you can't see, who is behind the camera, is the data manager. And as you can imagine, collecting medical records from 700 individuals for their lifetime is a lot of data. And so we are abstracting medical records, and collecting our own data, and all that goes into a very large database that David oversees. And then, of course, we have two administrative staff, Gloria and Linda, who help with budgets and all the paperwork kind of stuff that has to happen. We also have a huge number of collaborators. At the moment, all these people are volunteers. There was a time where we paid parts of salaries when we were really focused on very specific investigations. And because there are 263 genes on chromosome 18, some affect the heart, some affect bone, some affect muscle, many affect the brain. Any one person doesn't have all those problems, but in the group, we have a huge range of issues. So our collaborators come from neurology and immunology, ophthalmology, ENT, all medical specialties. And as we've learned more, some of our questions have gotten more specific. So we have collaborators that are expert on platelet function and bone health and much more specific questions, clinical questions. And then we also have mouse electrophysiologists and mouse behavior specialists, and there's Georgiana. So as we start to learn key genes, we're now trying to understand how they function, and you often need cell lines or mouse models in order to do that. And so we have begun to have those collaborators as well. Any questions at this point? Anything in the chat box, Holly?
No questions yet.
Okay, so I can show you our office. We have 3 office suites, and within across 3 office suites. So myself and Annis are here. We have an office suite across the hall and one down the hall with various people. And then let me take you down the hall, around the corner, a little ways to where we have 2 labs, and we'll go into one of those labs.
So as Dr. Cody's going down to the labs, let me kind of give you a couple of pieces of information. Is that we have over 700 people that are participating in the research, which is kind of like a number that grows every year. And then just so you have an idea, we have about 4,000 families that are members of the registry within the US. We have about 300 within Canada. And then we have about another 1,700 around the rest of the world. So just to kind of give you an idea on kind of the numbers of people that we kind of interact with. But as Dr. Cody has arrived in the lab, I'm going to pass it back over to her.
I can't hear Neil anymore. Did I lose the sound, or did I lose the sound, or are you not talking?
I can hear you.
We can hear
You
you.
can hear me?
Yes.
Somehow I just can barely hear anybody. So,
Noise.
what?
The noise in here.
Oh, the noise. Oh, yes. So apologies for the noise, because we've got an exhaust hood, a couple of there's lots of exhaust noise in the lab, background, white noise all the time. So that makes it a little more difficult to hear in here. We have two labs. This is our little lab, the tissue culture lab, which is really more interesting, I guess, to look at. Oops, I didn't put my glasses back on. I guess if I don't have my mask on, I can wear my glasses. Yeah. So did I just do the wrong thing? I wanted to. So what we do when people enroll in the research, first they have to send us, of course, their chromosome report that proves to us they in fact have a chromosome 18 abnormality. They have to send some medical records so that we begin to understand what their medical and developmental issues are. And they have to send a blood sample that we need to get within 3 days, which is often a limiting factor. And we ask for blood from the child and both parents. And the parents, in any analysis we do in the parents is just to compare to the child. So let me show you what we do. And anybody who sent blood, you'll get this blood kit with all these tubes. we ask for a lot of tubes because we do 3 different things with the blood. The first thing we do is isolate DNA. And let me see if I can show you the DNA here. Can you see the string floating in there? Tell me if you can see it. See the white sort of.
We can see it.
You can see it.
Yes, we can see it.
There we go. Can you see that?
Yes.
Okay, that's real life DNA. This is the second to the last step in DNA isolation. When it's the last step, it dissolves so that you won't see it like that anymore. It dissolves, and that's so as I said, this is second to the last step, the wow step. So what we do with the DNA, now that's one of the 3 things we do with the blood is isolate DNA. And what we do is microarray analysis, and we have high resolution microarrays that really focus on chromosome 18. And what this slide shows is a screenshot from the software.
Oh, they normally pop it up
Oh, they do.
so that they can see close. 'Cause
Oh,
I
okay.
can't really get close.
Oh, but then I'm pointing with the mouse, so that's not helping much.
How many?
So anyway, it's sorry, and tell me if you can or can't see things. So,
It's on the
so
screen right now, Dr. Cody.
this is I can't hear that. I'm sorry. What?
We have it pulled up on the screen right now too, enlarged.
Okay. Are you straight in front of that?
I'm not really straight. in front.
Okay, so this is just a partial screenshot from the software in which we do the DNA analysis. So because it's software, you can zoom in, you can move around, you can zoom in, zoom out, and it of course would have all the chromosomes. In this particular picture, it just has chromosome 18. And you'll see all these red and black and green dots. And every one of those dots is a data point. And all the ones aligned down the 0 line, here's the 0 at the top, all those aligned there and that are mostly black indicate the person has 2 copies of chromosome 18. Where they divert, and you can see it's very noisy. It's a very noisy technique, but the analysis looks at, only analyzes 3 consecutive data points to call a deletion or duplication. So you can see down here at the bottom, all those dots, or most of those dots are green, and the line is diverted to the left. That means that the person has a deletion right there. And what I forgot to tell you, though, is the way we do all those, get those data points is the microarray analysis. Slide stuck. Here we go. Okay, and that analysis is done on a microscope slide like this. And if I hold it just right, I can barely see it, and so there's no way you're going to see it. but there are arrays of DNA manufactured onto this slide that have about 250,000 known DNA sequences that are manufactured on here. They're from known positions on all the various chromosomes. And we put the DNA on here, and it takes about a couple of days to do that analysis. And it's read by a laser. And then the slide I'm showing you is the analysis that we get. And one of the really important things is this has, I think there's 10,000 spots on chromosome 18. There's a lot. When I first started in graduate school, the data for each one of those spots took 3 days to get. Now this whole thing takes 3 days, and it analyzes all the chromosomes, not just 18. So when this was invented, I felt like I died and went to heaven, and all the data that I had worked so hard over, 10 years to collect was just subsumed by 1,000-fold better data in a matter of months. So this is a very powerful technique. And as I said, you can zoom in. So here's a zoomed in shot. So on the left, or on the right, is a zoomed in view of what's in the box on the left, that blue dotted box. And in here, you can see, if you see these gray bars, each of those gray bars represents the placement of a gene. Now, in reality, the genes are in single file, one after the other, but they're spread left to right. And single file mean up and down in this picture. They're arrayed left to right just for readability, so you can see the names, because this is actually not a very zoomed in view, even though it's zoomed in compared to the other. But you can see here exactly where this person's deletion begins. And so this gene here is not deleted, and this gene is. So all the genes in this region, this gene, these are all within their deletion. And the genes at the bottom of the chromosome are not missing. And so what this kind of analysis shows us is how many copies you have of each section of the chromosome. What it doesn't tell us is there a rearrangement? Where are these copies? So this little piece down here at the bottom, we don't know where that is. Is this piece missing from the middle of the chromosome and this is attached up here? Or is this piece off, stuck on another chromosome? And we don't know that from this kind of analysis. So we have to employ another technique called FISH. Are you making them dizzy or what?
Probably. Probably.
Not a professional cameraman.
Yeah.
So that FISH is fluorescence in situ hybridization. And for FISH, we do a couple of things. There are fluorescent dyes that are specific to specific chromosomes. And so we take cells from our sample of blood that we get. We isolate the white blood cells that have the nuclei, and we do a chromosome preparation on a microscope slide, and then put this fluorescent dye that's specific to chromosome 18 on the slide. And here you can see all the chromosomes from one cell, and here are 2 chromosome 18s with the fluorescent dye here and here. Now, the important thing to note is there's no chromosome 18 material anywhere else. So that bit of 18 that you saw from that person's chromosome 18 is actually attached. And it's probably this one. This one looks shorter here, but you can't be sure. So that person probably has an interstitial deletion. And in order to be completely sure, you could do another FISH analysis. And this one uses 2 probes of 2 different colors. One that's green that shows you the centromere or the waistband part, the constriction on chromosome 18, and that's green. So in this slide, you can see here's a chromosome 18 and here's a chromosome 18. And then there's a red dye, fluorescent dye, that's specific just for the very end of chromosome 18. And you can see it's here and here. And so what this tells us is, in fact, is that the end of 18q is in the same chromosome as the centromere for 18q in both cases. So, whoops, that person does in fact have a interstitial deletion of 18q. So that's the second thing we do with the blood. So why we need so much blood. Then the third thing we do with the blood is to isolate the cells to grow in culture. So we put the blood in these expensive tubes, put it in the centrifuge over here, spin it down. The red cells go to the bottom. The white cells are in this little buffy coat up above. And we take those cells and we put them in a tissue culture flask along with culture media, which I realized I forgot to get out of the refrigerator, but it's just red liquid. Put that in here with the media and the cells. And we do one more thing. We put in some Epstein-Barr virus. And if we didn't add that virus, those white cells would double a couple of times and they would die. We could add the virus to immortalize some of those cells. And then they keep living forever, and we can grow them up, freeze them down, and then we have a stock of cells if we need more DNA, or if we need to do gene expression studies, or now, something we never dreamt about when we started this and started collecting these cell lines, is that we can actually create pluripotent stem cells from these and then differentiate them into neurons so that we can understand how the chromosome 18 genes are different in neurons of people with 18q deletions. Very exciting stuff, which is one of the things we're working on. And in order to grow the cells, they have to be at body temperature, because these are human cells. And so here's our incubator at 37 degrees centigrade. And here are some cells, some of those white blood cells, which maybe you can see floating in there. It looks kind of cloudy. Patty fed these just the other day. And one of the things, and there's an art to this, Patty's expert at it. What?
Move
Up,
back a little bit,
back a little bit. Okay.
and then tip it forward.
I don't know if you can see that. There are little bitty clumps in there. It may just look cloudy at this point. So the media, like I said, is red, and it has a pH indicator. So as the cells grow, they use the nutrients in the media, which then changes the pH and changes the color. And so now it's kind of orange. When it gets a little more yellow, then it's time to feed them. So this is an art. Every day you gotta check, judge the color, look at them under the microscope, decide if you want to feed them. And the way you feed them is you don't do what I just did. You actually let them sit. So all those cells go to the bottom. And then you take 10 milliliters of media off and add 20. And another 3 or 4 days later, you might take off 20 and add 40. And doing that over and over and moving them to a bigger flask, we eventually get enough cells that we can freeze them down. So let me put these back. And if you were here in real life, we would be able to show you what they look like under the microscope. But here's a picture of some cells, lymphoblastoid white blood cells that are grown in culture. And you can see they make big clumps like this. And the cells in the middle can't really reach the nutrients. And this is actually what you're seeing, is the way you see the cells. You're not seeing single cells, you're seeing the clumps floating. But the cells in the middle can't get the nutrients. So that's why it's okay to sort of shake them up every once in a while, or stir them up kind of like I did. Helps break up those clumps into all these single cells that you see here. And so here's just 2 images of what those cells look like. And as I said, there's an art to this. When you look at them under the microscope, they look a little darker outlined here than they do in real life. They look like little green grapes. And if they look like green grapes and they're growing in these clumps where you can see, all the grape shapes off to the side, that means they're happy. If they look like raisins, you're in trouble. So if you were really here, we could show you under the microscope, but can't do that. As I said, once we grow them up and we get a lot, then we spin them down in the centrifuge, put them in a very special freezer media, and we freeze them down for storage. And so let me show you that. When I skip. And when we're growing up the cells, all that work has to be done in the hood, which is sterile. There's it's a laminar flow hood. There's an airflow curtain right here, keeps air from the room from going in there. Everything in there is sterilized. And so when you open up that flask to take out media and add more, all that's done underneath this hood. Because bacteria double every 20 minutes, human cells double every 24 hours if they're happy. And so if you get one bacterium in there one day, by the next morning, that culture is overgrown with bacteria and your cells are dead. You got to make sure you get no bacteria and no fungus or anything in there. So all the work has to be done sitting in front of this hood. And as I said, when we go to freeze them down, we freeze them in one of these tanks. And every person who's enrolled, the affected individuals and their parents, we freeze down at least 3 vials of their cells, and they go in these little cryo vials like this. And the cryo vials go in a cave to be frozen or a box to be frozen, depending on which tank it goes in. And every individual has cells in every tank. That in case one of the tanks springs a leak, they're just giant thermoses with liquid nitrogen. If one of the tanks springs a leak, then you lose everything. We haven't lost everything because we've got 2 backups. So let me show you, I'm going to use the little tank just because it's easier to deal with. So this is full of liquid nitrogen, so it's very, very cold in there. And it has 6 little canisters. And each canister has about 30 of these cans, and it's really cold. Burn my finger. But you can see, I don't know why. It's colder today. Anyway, you can see we have lots and lots of vials of cells in this tank, and then these tanks, of course, are bigger. And we just got funding, or the registry just got funding to buy another tank to replace this really ancient one, which we got handy down from somebody. And it's not as big as the other 2, so we'll get another big tank. Let's see, what did I forget? Anything? I think I wish you were here in person so you could really look under the microscope and see what this is, what it's really like. But this is the next best thing. Do you have any questions so far about the lab stuff?
Yes, Dr. Cody, we have a question. We have a question. How much does the DNA slide cost?
Oh, not to say that. That microscope slide that I showed you with the array, the 250,000 manufactured DNA spots, that's about $600 some just for the slide. Then you need all the reagents that are proprietary, gasket, and all the other things to go with it. So it's quite expensive, but when you think about the amount of data you get, it's relatively that it's cheap because it's just revolutionary the amount of information you get.
Great, thank you, Dr. Cody. And Dr. Cody is going to make her way back to the office. And once she gets back to the office, she's going to be able to sit down and kind of answer some questions that you might have. Is that I would recommend that anybody, this is a unique opportunity to kind of, ask Dr. Cody any questions that a lot of people don't often get. So if you do have any questions, just, please don't hesitate to reach out. So just a couple of kind of financial things that whilst Dr.
Here you go.
Back, and thank you, David, for doing all the camera work, is that he actually it kind of does a number of different jobs that we ask him to. So thank you for that, David. But to give you an idea is that in an ideal situation, we need about $500,000 a year to keep the research center fully funded. At the moment, we're giving the clinical research center about $300,000 a year. So we're not up to where we want to be. Obviously, last year with COVID and all of our events closing down, things kind of got a little bit tighter. But we're hoping that this year, towards the third and fourth quarters, that we'll start having some events again, and we'll really kind of start be able to kind of push the fundraising back up to get to a stage of where we need to finance the clinical research center. We are the only clinical research center in the world that is doing research specifically on chromosome 18. And as I mentioned earlier on, we're celebrating our 30th anniversary coming up in actually September. So Dr. Cody has had numerous publications in the scientific community. One of the great resources that hopefully she'll talk about is the clinical management guides. And the clinical management guides is something that kind of every family should absolutely download and give to their physician. But hopefully Dr. Cody will be joining us in a second. She normally doesn't take that long, but perhaps something happened to her computer.
Oh.
Here she is.
Oh, I clicked on the video and it said that I couldn't do it because of the host or something. So I guess
okay.
I undid whatever you did.
Great. Well, I'm going to pass it over to Dr. Cody for any questions. So Holly?
Sure. So we have a question. I know Neil talked a little bit about it, but why did you start the Clinical Research Center?
Oh, well, Liz, here's my Liz. She's 36 now. When she was born, what little was known, we were of course told there were 65 other families in the world, but what little was known really had to do with how you might recognize somebody with 18 cumitis, like, a broad mouth or low set ears or, things like that. And it didn't really say whether their ears worked or not, or, it did say they lie in a frog-like position in a vegetative state and have a normal lifespan, which was not particularly helpful. And so most of the direction we got was wait and see, which is pretty unfulfilling. So we needed to learn more. And as we learned more from Liz, then I thought we've got to, see what other families might be out there and what we can learn from them. That's a condensed version.
Awesome. Does anybody have any questions? Donna, Tara, anybody else? We did have another question, and if you guys have one, you can feel free to unmute yourselves or type it in the chat box. So Dr. Cody, how does the research help individual families and the chromosome 18 community as a whole?
Well, so appreciating that most clinicians don't necessarily even have access to, and especially not the time to go look up 50 papers and figure out what applies to their patient and what doesn't. We converted everything to a physician management guide, so they're all in one place. And even when you write a review article for the medical literature, it's not ever organized in a way a clinician can use it easily. So we organized it not for publication in a journal, but for usefulness. And so we have patient management guides, originally published in 2016. We did a big update last year. We're kind of at the point where we have enough people that it you know, it takes a few more years to get enough data to move the needle just a little bit in the percentage of this and that. If something new develops, we'll of course update them right away. So, and all that information's free. So anybody can get it anytime off the Research Center website. For the families enrolled in the research, there are a couple advantages, I guess. One, they know us, we know them. They ask us questions all the time. We get lots of emails and phone calls from the families involved. And additionally, one other thing went in and out of my head. So the other way being involved in the research is helpful is, for example, there's actually multiple examples, but to pick one, we learn about the function of a gene that we think would affect somebody who has a deletion of that gene, but the symptom might not be obvious, or it might be adult onset, or it might be something that takes special testing that could cause a problem later on if you didn't know about it. So if you're involved in the research, you're going to get a survey that asks you questions or asks for a specific sample of something or a test or whatever, because you'll get a letter saying we're asking this question. And so it's not even a result yet. It's just, this is a question we're asking. So all the families in the research know already this is a potential issue I need to think about. Families not in the research probably won't learn about it for 2 to 3 more years, because that's how long it takes. We first survey our families to find out what they know or what information we can get from medical records that might be hints. We get additional testing, then we have to write it up, and then it gets published. And the timeline from submitting a paper to actually having it in press, in where people could get it even online, is usually about a year. And so all that just takes so much time. And I got a frustrated email a year or so ago from a dad whose daughter had some issues, and as a consequence of some surgeries that she had they been involved in the research, they probably wouldn't have done that surgery, or they would have made special accommodations. I mean, because of this surgery, she had side effects that were not related to the outcome of the surgery itself, but were an outcome of having had surgery. And they probably wouldn't have had that had they or done it quite the way they did had they been a part of the research. So I'm sorry, I can't do anything about that. I just, you know, you can't tell people what you're thinking about until you have data. And really, so you're in the know, you're in the inside group if you're part of the research. So, and sometimes we have asked questions of families and found out that in fact it is not an issue. So, and just likewise, the families involved in the research find out that one, we're worried about it, so they get worried, but they also find out first that it's not really an issue. So.
Wow, that's really interesting. So it does seem like it's really valuable to the families to be a part of the research, and then also for the clinical research center to have that information.
Oh yeah. Well, and the other part of that is, I really hate Facebook. Probably shouldn't mention that, but I do. And so when I go on Facebook, the only thing I really ever read are things about Outlander. So that's all I ever see now when I do look. So that's really good. I just don't get sucked into anything. But every once in a while, I find out something from somebody else, or I see something on there where somebody says, does anybody have this problem? My child has this. Well, you know what? They're invisible to the world, except for those few people who happen to see that note on Facebook. If you're involved in the research, that's information we collect. We get 4 people or 3 people with this condition who have this unusual problem. Then we start asking questions about it, and we have all these people enrolled that we can get some real answers instead of just from one person who happened to read your post answer, who actually didn't know what they were talking about. So so it's important to be in the research in that regard. I mean, we've had individuals that we hear about who passed away. They weren't in the research. We have no idea. They'll say, oh yeah, they had 18p minus and they died from this, but we don't know whether they had a little bitty deletion or a big deletion, or they had some other complex rearrangement because they weren't in the research. So even knowing the cause of death after they've died doesn't help us because we don't know how to compare that to everybody else. And so they were invisible to the world. And I don't think anybody really wants to be invisible. It's just they don't understand how important and how visible they can be and how important they really are to everybody else with their child's same condition.
Wow. And I did pop the link in for the enrollment and for the clinical management guides. So definitely people on the call, go ahead and take a look at that when you can. But you really taught us a lot about why it's important to be a part of the research. So thank you. Does anybody else have any questions? We do have one more. If you received additional funding, what would be next for the Clinical Research Center? Like, is there a wish list or?
Well, you know, it all depends how much money you're talking. Today, we had a minor crisis because the computer that I showed you in the lab died. And it's essential for running a piece of equipment that now we can't use because the software to run that was so out of date. It had to be on that computer that wasn't getting updated. In order to get updated software, you have to get the new equipment, and that's $20,000. Anyway, so that's today. We'll try to shoestring things along somehow so we can keep using that. So, but it'll be a temporary fix. So, I mean, all the pieces of equipment, and that piece of equipment only measures the concentration of DNA in a tube in one of our samples. But you got to know that. You can't do anything if you don't know that.
Right.
So in a way, in the world of equipment, of scientific equipment, that's just like a trivial thing, but it's $20,000. So if we actually had a lot of money, what we really, really need to do is we need to bring in and do comprehensive clinical evaluations like we used to do on not everyone who enrolls, but key people. We have people with really unique deletions. Some are really small. And so if we knew what exact manifestations they had of that really small deletion, that would really help clarify what genes in that region are important for everyone with bigger deletions that involve that region. So we've got some really, some people who enrolled with really unique deletions or duplications. We also been doing this 30 years. So now we have adults. We don't really know a whole lot about what's going on with the adults. And if they develop adult onset conditions, we have to just rely on them telling us about it. And by the time they're diagnosed, they've already got it. And so some conditions can be helped if they're caught early, but we don't know what to tell people to look for if they could catch early. So I would really love to do more clinical evaluations. That's just key. And the other sort of big picture things is mouse models and molecular analyses. We're just writing a grant with 2 collaborators in other departments here, one a mouse person, one a neurobiology person. And we're writing a grant to the National Institutes of Health. It's really only a pilot project grant, so it's only $75,000 for 2 years. But hopefully that'll get us the data we can apply for a bigger grant. But that involves one specific problem that we're looking at. And so there are others where we could involve other collaborators to look at cellular aspects now that we're starting to understand which genes, and mouse models, so mice with deletions of single genes. That could help us understand, because you can dissect a mouse brain and learn about its structure and what's not right if it has a deletion of very specific genes that are on chromosome 18. wouldn't want to do that to people. So it's an essential step. I know we do everything we can to use cell lines and organoids and such things, so we don't have to rely on animals, but sometimes you have to. And so even though we try to minimize that as much as possible, partly because it's very expensive, partly because we just don't want to do that, it's still an absolutely essential step. You can't get anywhere without it. So you can't get to drug development, I'd say, without it at some point. So so there's lots of molecular sort of avenues that we could pursue with more funding. So lots of things. I mean,
Yeah.
There's even with 263 genes, and only we think 10% are responsible for the problems, but still a lot of avenues to pursue. And it's a lifetime of clinical information about people that we don't know enough about. We don't know the natural history, is what the scientists would call that.
Well, thank you so much, Dr. Cody. Thank you, Neil. Thank you, David. Dr. Cody, I did want to read a message from Tara. She said, thank you, Dr. Cody. It's very interesting to see the lab. We appreciate your time. We definitely thank you for your time and all that you do. Does anybody have any other questions or anything they want to add before we end our time today? Don't everybody talk at once. No, I'm just kidding.
Well, thank you, everybody, and I apologize for our unprofessional sort of cameraman make-do arrangement, but
No, it was great. Thank you so much.
So
Thanks, everybody. And please, if anybody does have any questions that you think of afterwards, don't hesitate to reach out to Holly, and she'll get them over to Dr. Cody. So thank you very much indeed, everybody, for joining us, and hopefully we'll see you soon.
yeah, hopefully in person in the not too distant future.
And we got another message. Tara said it was a great tour. Thank you for all the information. And I put a few links in the chat, so I'll go ahead and follow up with an email so everybody has the links. And just thank you so much for your time and joining us today.
Okay. Nope. Happy to do it. Thank you.
Thank you.
Bye.
Bye. And Donna said thank you too.
Oh, hey, Donna.
Bye. Have a good day, everyone.